Hello, everyone. My name's Dr. Paul Richardson, and I'm the Clinical Program Leader and Director of Clinical Research here at the Jerome Lipper Multiple Myeloma Center at Dana-Farber Cancer Institute in Boston, Massachusetts. And I also serve as the R.J. Corman Professor of Medicine at Harvard Med School. And it's really my privilege to talk to you about today the emerging role, in my view, of CELMoDs in the treatment of relapsed refractory myeloma in particular, and to touch on why this is so important as a vital new therapeutic option, in my view, for our patients, in particular for relapsed refractory disease, and potentially with exciting new opportunities in the newly diagnosed setting, as well as in maintenance, specifically for the CELMoD iberdomide. The other parts of my conversation are going to be focused on the relapsed refractory setting with mezigdomide. Now, why are new treatments for myeloma needed? Well, I think the most important thing is to understand that myeloma truly is not just one disease, it's multiple diseases. And in that same context, it's highly complex at diagnosis and particularly at relapse. And so thus, one size truly does not fit all. And we face many challenges in that setting, not only mutational burden, immune exhaustion, infectious complications and myelosuppression, but very importantly, extramedullary escape, which can be so challenging. And I illustrate here in the center of this slide with liver involvement in a particular patient with extensive extramedullary disease. And this mutational burden is best summarized in these whole genome sequences, which show remarkably high mutational burdens of relapse, depending, also driven to some extent by prior therapy, which then can result in worse outcome. The good news is that we have a whole array of new drugs to target all of this, as well as very exciting established agents that have approval broadly across the globe, with some differences existing in that regard, but nonetheless, a broad array available of not only immunomodulatory treatments, proteasome inhibitors, monoclonal antibodies, acetylators in certain healthcare jurisdictions, not others. And then obviously in the more recent spaces, antibody drug conjugates, targeted therapies, which I've summarized in that column. And I think most excitingly, CAR-Ts and bispecifics, recognizing that we've just had the recent approval of linvoseltamab in the US, which I think reflects really the momentum behind this. And in that context, my emphasis on the CELMoDs is boxed in green with both iberdomide and mezigdomide, and we'll talk about those in more detail. Now, how does this extend to the US? Well, most importantly, in the US, we currently have a selection of options and guidelines, and this is derived from an important paper from the Journal of the National Comprehensive Cancer Center, so the NCCN, in which a very nice summary is presented. And in that context, I also wanted to add these later lines at relapse of ide-cel and cilta-cel, but obviously, of course, to emphasize the emerging role of the bispecifics, including linvo in this setting. So as we think about the sequencing of therapies in relapsed refractory myeloma, I think it's also important to share with you the challenge of post-anti-BCMA therapy. Where do we go? And here I think is at a point of very exciting emphasis for the potential role of CELMoDs, and we'll talk a little bit more about that in a moment, recognizing that the CELMoDs may have an important role earlier. And then finally, just to give you some context, I want to touch on what would we expect in terms of outcomes in the relapsed refractory setting. And one of the most important early studies in this regard was published a few years ago now by Dr. Gandhi and colleagues. And what they showed was that once you're CD38 antibody refractory and you've had both proteasome inhibition and immunomodulation, the outcomes are really very challenging. And that median overall survival is around 9 months at this time. But as you can see, the more classes of drug you've become refractory to, no surprises, the worse the outcomes become. And this is particularly true when we think about both post-lenalidomide and CD38 monoclonal antibody therapy. And this is a more recent paper published by the lead author, Dr. Kartik Ramaswamy, where I think it's actually very important to note that from very relatively large databases, including Preamble, Connect, and also the Flatiron Health database, which was almost 900 patients strong, that you can see that the outcomes for triple-class refractory patients remain very guarded, even despite the advent of exciting new therapies targeting BCMA in more recent years. So in that setting, how do the CELMoDs fit in? Well, I think most importantly, it's critical to recognize that they are not the classic IMiDs, in my opinion. They are that much different. They're true degraders, and they target Ikaros and ALOS, and they do so with targeted protein degradation that's particularly profound. And this is highly synergistic with other classes of drugs including both proteasome inhibitors and, of course, antibody therapies. And with that in mind, of this degrader class, it's important to note that not only do they have a direct effect on the myeloma cell and this closure of the cereblon E3 ligase complex, but they also generate downstream remarkable effects on the immune system, including programmed cell death from the myeloma itself. Now, to help distinguish the immunomodulators from CELMoDs, I just include this slide, which helps you understand that the molecular structure, while they share certain commonalities, have distinct differences. And this sets, in my view, the CELMoDs apart. And what does this mean in terms of their activity? Well, I think this, to me, is the most striking. You can see that cereblon binding is that much more dramatic, that targeted protein degradation is also magnified. The effects on tumor proliferation, apoptosis, and above all on immune stimulation are profound, and there's great synergy then in combination. So I think this is a very informative construct. Now, what's the preclinical data to support this? Well, I show that in this next slide, where we have really important evidence of both iberdomide and mezigdomide synergizing with bortezomib, synergizing with steroids, and most importantly, the degree of cell kills do appear particularly more profound. Understanding this is summarized in this slide. This is comparing lenalidomide and pomalidomide and the engagement of the cereblon E3 ligase pocket by the respective molecules. And as you can see, there is a larger molecule in interaction by virtue of iberdomide that results in a dramatic increase in the degree of Ikaros and ALOS degradation, reflecting this closure of the E3 ligase pocket that's even more profound when we look at mezigdomide. And this, I think, is really exciting data derived from patients from our work in the 001 study for mezigdomide, so-called CC92480, and for excellent correlative work also from the iberdomide team for the CC220 study, in which the use of iberdomide and dexamethasone in relapsed refractory disease was studied. And what you can see here is that there is tremendous effects on the immune cell microenvironment that appear favorable. And so overcoming T-cell exhaustion becomes a critical aspect of the activity of both mezigdomide and iberdomide. So to summarize these, I think there is cellular effects that are profound, there's activation of immune components that's essential, and also there's dramatic effects on cytokine production and cytotoxicity, all of which appear favorable. And recent data from the ASH meeting last year and EHA this year have continued to build on this story and emphasize the value of these agents in immune exhaustion. So what are we looking at clinically? Well, let me share with you first our work from the mezigdomide dexamethasone study in relapsed refractory disease, the so-called 001 trial. And here in the dose escalation phase, we treated almost 80 patients, and then we dose expanded at RP2D, over 100 patients with heavily pretreated disease who were in very high risk, with 40% with extramedullary disease, almost 40% with high-risk cytogenetics, a median of 6 prior therapies, and very importantly, a significant number of patients who were BCMA refractory. And what we saw was an encouraging median DOR for an all-oral regimen of about 8 months. The median PFS, which obviously is represents both responders and non-responders, was around 4.5 months. And I think particularly exciting to me was the duration of response seen in those patients who received prior anti-BCMA therapy, exceeding 6 months at almost 7 months, and the median PFS, which reflected that as well. And then when we look at the actual response rates, and remember, this is just an all-oral regimen used to good effect in the era of the pandemic, when that was reflected by the very low rates of serious complications. We were excited to see these response rates. Overall, 41% for simply an oral regimen in triple-class refractory patients, the recommended phase 2 dose. And then in those patients with extramedullary disease, we were very gratified to see a response rate of 30%. And then excitingly, in those patients who had had prior anti-BCMA therapy, the response rate was 50%. Now, what about the EMD story? Well, in this regard, we had some very interesting results. This is a patient from Canada in whom their extramedullary disease, a large lesion in the liver, literally responded beautifully after 2 cycles of mezigdomide and dexamethasone in a sustained fashion. And this is a patient actually of mine who presented with incredibly high burden of extramedullary disease, closing off his porta cath, in fact, so my nursing team could not access it, on his chest wall. And as you can see, after 4 months of treatment, which he tolerated remarkably well, and despite being infected with COVID, from which he fully recovered, fortunately, he had a remarkable response. And you can see that illustrated in the PET CTs. So with that in mind, what about building on the Mezidex platform and adding to it with proteasome inhibition? Well, this data is summarized beautifully here, led by my colleague, Dr. Oriol. And what you can see here is that the combination of mezigdomide combined with either proteasome inhibitors in the form of bortezomib, or for that matter with carfilzomib, generated remarkable response rates. I want to bring your attention to this cohort of patients in which response rates ranged between 85 and 91%. Which we thought was particularly striking, as these were patients all previously bortezomib exposed. And with that in mind, the combination then of this platform with antibodies in the form of daratumumab or elotuzumab were equally favorable. And I want to actually point out to you the response rate combined with daratumumab in relatively early relapse, 75% response rate. And even for those patients getting elotuzumab after daratumumab failure, the response rate was a respectable 50%. So as we summarize the data, you can see here the grids summarizing those data sets. As I mentioned, 41% response rate for mezigdomide and dex, with particularly exciting signal in both EMD and BCMA refractory patients. And then in combination, when you combine with bortezomib, the response rates that range from 75 to 91%. With longer follow-up, we saw an encouraging response rate from the combination of mezigdomide and carfilzomib at 85%. As I mentioned just now, combined with daratumumab, it was an encouraging 75%, and with ELO approaching 50%, it is in fact at 45%. Now, what about novel combinations? Well, I think there are a number to share. One of the most exciting approaches is summarized in this slide, where under the leadership of Dr. Luciano Costa, we've been able to conduct a very innovative study combining mezigdomide and dexamethasone with two other rational classes of drug, in particular the targeting of the EZH2 pathway, and also very importantly in patients with 1q amplification and overexpression of CKS1 beta, the goal of seeing if we can also have an impact on the RAS-MEK pathway, particularly with the drug trametinib. So with all of this in mind, so far we've seen some interesting results. As you can see, interesting safety across the board. But when you combine mezigdomide and dexamethasone with tazemetostat, you can see that response rate is robust at 50%. When you combine mezigdomide and dexamethasone with trametinib, it's a remarkable 75%. Now, obviously, these data continue to mature as we expand dose cohorts and continue the study. But I think we're all very excited about this, particularly given the high rates of T-cell redirection. One of the most important observations has been that with the bromodomain inhibitor, BMS 986158, the response rate has been a bit more modest. And so I think in that context, whilst these are a very sick and a resistant population, nonetheless, that particular arm is not being taken forward. And the combination that is being explored further is mezigdomide, dexamethasone, and tazemetostat, and mezigdomide, dexamethasone, and trametinib, which obviously is exciting. Now, across the board, there are a number of very exciting other efforts underway. One trial that I think is particularly interesting is being led by colleagues in South Korea, and this is the combination in the Asian Myeloma Network. This is the combination of mezigdomide and elranatamab, and I think is a really exciting construct with data eagerly awaited in this regard. We also have, obviously, great work with iberdomide. I want to touch on this in the interest of time relatively quickly, but suffice to say, in the relapsed refractory setting, under the leadership of Dr. Sargalonio, we've been able to demonstrate an encouraging response rate at around 26%. And then in the recommended phase 2 dose group, it's a little more robust at 36%. But recognizing in this context, these responses have been durable. And I think what's interesting in this particular analysis, if you look at cohort D, this is the large cohort comparable to what I showed you with mezigdomide and dexamethasone in terms of size. And you can see here the response rate's about 26%. And these patients are, of course, heavily pretreated and relapsed refractory. If you look at the cohort 1 as part of that, cohort I as part of that, the 38 patients strong, these are BCMA exposed, and we see a response rate around 36.8%. So remembering with mezigdomide in the same groups of patients, we saw a response rate of around 41% for the group overall. And with mezigdomide in the BCMA exposed patients, we saw a response rate of around 50%. It's very interesting that these platforms are similar in the sense that we clearly see responses, but marginally less so with all the caveats of cross-trial comparison. And we would expect this, given that iberdomide is less powerful in terms of its cereblon E3 ligase closure and its targeted degradation and the mezigdomide. But nonetheless, very gratifying to see both drugs, both iberdomide and mezigdomide, perform very well in this setting. Now, recognizing that the goal is to move iberdomide earlier into the treatment of multiple myeloma, this data, I think, are reassuring in the sense that showing nice synergy and combination. Again, the degree of responses do appear a little bit less than we see for mezigdomide, but again, very much as explained. And then combining iberdomide with ixazomib as an oral proteasome inhibitor and cyclophosphamide dexamethasone as another one in the relapsed refractory setting, great to see solid response rates and excellent tolerability. So this is what we're looking at in terms of phase 3 studies of the CELMoD triplets in relapsed refractory myeloma that are ongoing. Excalibur has recently completed enrollment, and we're eagerly awaiting results from this. This was a trial comparing iberdomide, daratumumab, and dexamethasone to the control of Dara-VD. And this study, I think, will be very exciting and give us some excellent output, hopefully by the end of this year, if not in the first quarter of next year. Now, particular excitement are Successor 1 and 2. These are studies comparing either mezigdomide, bortezomib, and dexamethasone to the control in the setting, which is POM-VD, or mezigdomide and carfilzomib to the control in the setting, which was the doublet of carfilzomib and dexamethasone. Now, in Successor 2, patients had to be Dara exposed or CD38 exposed, and they could optionally be CD38 exposed in Successor 1. And we've been very pleased to see rapid enrollment to these trials. Successor 2 is almost complete, Successor 1 well on its way, and we're looking forward to exciting readouts in the relatively near future. Now, what about the upfront setting? And I want to touch on this very briefly. But iberdomide is being tested in the upfront setting because clearly, when used early in the disease, its efficacy is remarkable. Initial studies combining iberdomide with bortezomib and dex, presented first by Darrell White at both ASCO this year and at EHA in the summer. You can see here that the response rates are very encouraging, 100%, recognizing small numbers of patients. But what I'm particularly drawn to is the high rate of MRD negativity, which is basically already estimated at 50% and likely to improve over time. So with that in mind, iberdomide being developed as a newly diagnosed myeloma patient treatment of choice is coming forward in a variety of trials, with MRD as an important endpoint. It's also been explored as a maintenance strategy, and why this is particularly interesting is because of the absence of effect on C1k alpha, which means that by that it does not generate or does not favor p53 mutation in clones of disease. And that, of course, or clones of any kind of clonal situation, resulting in a presumed much lower rate of second cancers. So what we're hoping is that iberdomide will become a maintenance drug of choice because it's both well tolerated, exquisitely active, and it may have a much lower second cancer risk. So a number of trials looking at that with early data looking very promising. And then finally, it's worth mentioning that there are a number of post-transplant studies currently under study, which are showing a particular promise and just underway with data awaited, eagerly trying to improve upon responses in the longer term, with the primary endpoint summarized here. Now, with that in mind, I do want to quickly do a shout out for the Determination 2 trial being led by my colleague, Dr. Clifton Moe. And what you can see here is that we're looking basically at a very MRD-adapted trial, which is designed to assess the combination of isatuximab, iberdomide, and VD as induction remission therapy. And then in those patients with standard risk or MRD negative, stem cells are collected but transplant kept in reserve. And these patients continue on either IBA or ISA-IBA maintenance, depending on their MRD status. Conversely, for patients who are either MRD positive or high risk, they are able to then go either to high-dose melphalan or a fixed duration of bispecific, and in this case, linvoseltamab, which I think is so exciting, followed by maintenance. So a really exciting, innovative design, and really give enormous credit to Dr. Clifton Moe in his innovative ideas around how to build the design of this trial. And this study is currently in the process of being opened across the United States and should be, I think, very exciting. As I mentioned, the potential benefits of iberdomide center on its ability to have lower rates of infection and at the same time reduce second cancer risk. So in summary, we've got a very exciting field in terms of the CELMoDs, with phase 3 trials of mezigdomide and iberdomide ongoing in the relapsed refractory setting. And I'm very excited to see these emerge. And then at the same time, we have upfront opportunities for iberdomide that are really, I think, very promising. And in that context, the use of MRD, which has been recently validated as a tool for primary outcome in various settings and adopted by, or hopefully being adopted by the FDA shortly after a very positive ODAC earlier this year. So really exciting times in the field. And I think as we think about the evolving role of CELMoDs, I would summarize it by saying real promise for mezigdomide, in particular in the relapsed refractory setting, particularly in the setting of immune exhaustion, and also the need to combine with other drugs. And I'm very excited for the prospects of iberdomide in the upfront setting and as maintenance, but of course also in early relapse. And at that point, I would like to thank you and appreciate the kind time and attention, and look forward to next steps and future innovation in myeloma again. Thank you very much, everyone.